In mitosis, a diploid parent cell or a diploid mother cell, the DNA is replicated, and then two identical daughter cells that are also diploid are formed. In genetics, the thing that really is the basis for how alleles segregate, Mendel's first and second law, has to do with what happens in meiosis. Because in meiosis, you don't start with a diploid and end up with two identical daughters. You start with a diploid cell and you end up with 4 haploid daughter cells. So in meiosis, basically you start with a 2N diploid gametophyte is what it's called. It's like a parent of what are ultimately going to be gametes. So you start with a 2N gametophyte. It still undergoes interphase, and then in the S phase of interphase, the DNA is replicated. I like to say it's duplicated the DNA content, but you still have 2 pairs of chromosomes. So it's not really 4N, because 4N would suggest that you had 4 each of the chromosomes, and you still have 2 each of the chromosomes, but they have double the DNA content. So 2 times 2N. These are going to undergo 2 divisions. So meiosis 1. After meiosis 1, and what we'll find out is that the homologs separate. So the number of chromosomes is actually reduced in half, but they still have double the DNA content. So I'm going to call it 2 times N DNA here and 2 times N DNA in here. Now these they don't replicate the DNA again, they undergo a second meiotic event. So I'll just say meiosis 2. These go into meiosis 2, and what we end up with is that, well, the sister chromatids separate, and we end up with 4 haploid sex cells, or gametophytes. I'm not gametophytes, but gametes. So from 1 diploid gametophyte, we end up with 4 haploid gametes. Here's just a figure kind of giving a depiction. If you look at mammalian sex cell formation, you start with a primary gametophyte, which is a spermatocyte, and you end up with 4 sperms per parent. In the formation of the female gametes, we actually get these sort of inactivated polar bodies. We won't worry about that, but in general, if we think about it just big picture for our purposes, you would end up with 4 haploid gametes. The phases of meiosis are very similar. Well, they're called the same things as the phases of mitosis, except what happens in the first meiosis is where the difference occurs. So in meiosis 1, this is the first cell division. Remember, we had interphase, G1, S, G2. Now the cell enters meiosis 1, and it has double the DNA content. In prophase 1, so if you see a number after the stages, then you know we're talking about meiosis, because in mitosis, there was not 1 and 2, right? So in prophase 1, the chromatin condenses just like it did in mitosis. And you get your dyad chromosomes with 2 identical sister chromosomes. But something else happens in prophase 1 that does not happen in prophase of mitosis. And that is that homologous dyad chromosomes and I'm just going to call them dyads, homologous dyads pair up in a process called synapsis. So let's say that this was the one that was inherited from the maternal lineage, and let's draw a second one. From the paternal lineage, and I'll put some little stripes in here to indicate. So they pair up, they physically associate. And while I'm looking like an overlap here, I'm going to show you kind of a cartoon depiction of what synapses are more like. So here we've got our homologous chromosomes, sister chromatids held together at the centromere. Here's the homolog. They've got double the DNA content because the DNA has been replicated in S phase. Here's synapses. So there's actually protein complexes that form in between these non-sister homologs. So these are homologs to each other, but they're not sisters. So they're non-sister homologs. They physically associate, like they almost like zip up together. And so this process is synapsis. And it occurs during prophase 1. So my artwork didn't do it justice, but so essentially these would zip up together during prophase 1. Together, these two homologs, so these synapsed homologs, called a bivalent. And each chromatid within the bivalent, the chromatids, the sister chromatids that are in the bivalent form a tetrad. So the entire structure is the bivalent, the chromatids within the structure are the tetrad. These are sisters to each other. These are non-sister homologs. And another process, which I'm just going to put over here with a little asterisk, because we're going to put it on the back burner and it's going to be our topic for the next lecture, is that a process called crossing over occurs. So exchange between non-sister chromatids can occur. Exchange or a swap of DNA can occur between chromatids in a process called crossing over. And I'll go back to that figure again. So here it's showing that there's an actual physical exchange of the genetic information between some non-sister chromatids, and that's crossing over. Like I said, we're just going to put that on the back burner for now, but this is introducing that it occurs in prophase 1. So these are the major events of prophase 1. This did not happen, this pairing up did not happen in mitosis. So I'm going to put another asterisk here because this is different than what happened in mitosis. This did not happen in mitosis. Together, during prometaphase, again, the nucleus dissolves and the spindle fibers attached to the kinetochores at the centromere. So I'm just going to shorthand it, spindle fibers attach. And during metaphase 1, so this is prophase 1, prometaphase 1, and during metaphase 1, the bivalent, those paired up dyads, so the homologous dyads, or the bivalents, randomly line up on the metaphase plate in pairs. I'm going to say in pairs. And this is significant because if they didn't line up in pairs in mitosis, they lined up single file. So actually, let me try to diagram this out. What I mean by that, what I mean by randomly. Well, I'll save it for the end. I'll do a little sidebar over here. So I'm going to do a shortcut, make my homologs look like these, a little easier for me to draw. So they line up in pairs on the metaphase plate. So here's our cell, here's the equator. And what I mean by randomly is that if this is, say, the paternal chromosome, it can be on either side of the equator. but they line up in pairs. So maybe I'll put another little pair up here, and I'll put the paternal chromosome on the other side and the maternal on the other. So there's nothing that makes all the paternal ones line up on one side of the equator and all the maternal ones line up on the other. It's random. So this is a key sort of situation here too. In anaphase, remember the spindle fibers are attached. In anaphase, the homologous dyads are pulled apart. They're pulled to opposite poles. And then you get your, you get telophase and cytokinesis. I'll just put it up here before I change the screen. So you get telophase and cytokinesis. Where the two new cells form. So telophase cytokinesis will say the division of the cells is completed. And notice now we have one of each chromosome, but they have double the DNA content. They have identical sisters. Now these cells would double the DNA content, but only one of each homolog because we separated them. Now these are going to directly go into meiosis 2. So in meiosis 2, it's the same phases occur. And it's just like mitosis. So you have your you have prophase, where we get the condensation. You get the prometaphase where the spindles attach. So prometaphase 2, chromatin condenses, prometaphase 2, spindles attach. And just like metaphase of mitosis and metaphase 2 of meiosis, the dyad chromosomes line up, but they don't line up in pairs now. They line up single file at the metaphase plate. This time I'll use the term equator. Anaphase 2, the sister chromatids are separated. They're pulled to opposite poles. And it's important to understand that when sister chromatids are separated, they're no longer sister chromatids. They become chromosomes in their own right. So if I just kind of try to depict this a little bit here. So let's get our little chromosome, we've got a big chromosome, spindle fibers attached, lining up single file at metaphase 2. These guys get pulled apart. We're going to have telophase and cytokinesis. What we end up with here, when these get pulled apart, it's no longer a sister chromatid. They are only sister chromatids when they are physically attached at the centromere. So they get pulled apart, and then this one's going to get the same identical content. The sister chromatids get pulled to opposite poles, and they become chromosomes in their own right. So they were sisters when they were together, but once you pull them apart, they become chromosomes. And we call these chromosomes with just the singular content of DNA. These are monad chromosomes. And then telophase and cytokinesis. The division process is finished up. And the chromosomes will decondense, the nucleus reforms, and then you have the cells formed. Now remember, when we went into this, we didn't just have one cell, we actually had a second cell. That one had the little one was the solid and the big one was the dashed. Those get separated. Those got separated as well. And we have one little one, and we have one big one. So at the end of meiosis 1 and 2, we now have 4 haploid gametes. Let's take a look again at some cartoon figures to support this because my drawing's a little bit sloppy. Here is a nice little cartoon. I like this one. So before, this would be prior to S phase of interphase. If we imagine the chromatin as condensed, just so that we can visualize it, we have monad chromosomes, monad chromatin really, in the cell. So this would be before the S phase, before the DNA is replicated. After S phase of interphase, that all the DNA got duplicated. And so rather than just having one sort of set of DNA per chromosome, we now have two. And we call these identical sister chromatids. They're held together at the centromere. And these are homologs, and they pair up. They pair up, they physically associate. A process called synapsis, and crossing over can occur. After meiosis 1, these guys separate. Now we don't have both homologs inside the cell. And after meiosis 2, these sisters separate. And so what we end up with is half the DNA content that we had in the starting parent cell. So I like this as just kind of a nice big picture overview. Now the whole topic of this mitosis and meiosis was going to be the chromosomal basis for independent assortment and segregation of alleles. So it really comes down to putting alleles on these chromosomes really trips people up. So I want to diagram through it all. Now, you might want to try this before I diagram it to see how we match up. So diagram the alleles on chromosomes of an individual who is heterozygous for genes A and B, so a dihybrid. Gene A is located on chromosome 1, so a larger chromosome, and gene B is located on chromosome 10, a smaller chromosome. Diagram what the chromosomes would look like during metaphase 1 and depict the alleles on each. So pause the video, try to see if you can imagine zooming forward to metaphase 1 and what these would look like in metaphase 1. And I'm going to go to a fresh page and I'm going to diagram the whole process out from scratch. So we've got a dihybrid individual. So let's imagine what those chromosomes look like inside the cell before replication. So before the DNA is replicated. And we're going to make them look like they're condensed just because that's easier to visualize. And I think I am going to use colors. So we can use, we've got a blue chromosome. And remember, chromosome 1, gene A is going to be on chromosome 1. So here's chromosome 1, but we don't just have one copy. We inherited one from dad and one from mom. So here's chromosome, here's the homolog, chromosome 1. And then there's a smaller chromosome that will, this is like a nucleus. Inside the nucleus, if we would just visualize these chromosomes in no particular order, they're not paired up. So what happens after the S phase of interphase? So what happens after the S phase of interphase is all of these duplicate. So let's visualize now the duplicated chromosomes. And I'm just going to draw them like this. Little guy. Let's draw the homologs. Little guy. And let's go back here to this before replication occurred. So that chromosome, or that gene A, the locus for gene A was on chromosome 1. So let's make up a locus, and let's say the dominant allele was on the paternal chromosome, and let's say the recessive allele was on the maternal chromosome. They're homologs, same gene, same physical location, different variation of alleles. And it said that gene B was on a smaller chromosome, chromosome 10. Just for consistency's sake, let's put gene B on the paternal for no reason. I'm randomly doing this, and the recessive on the maternal. Now, after S phase, what are on these sisters? After S phase, what are on these sisters? These are identical. So after the DNA has been replicated, you have replicated all of these alleles as well. This one had the little b, and this one had the little b, this one had big B, this one had big B. Now let's make them synapse, and let's zoom forward to metaphase 1. So now I'm going to physically associate my homologs, and I'm going to line them up at the equator. So this one had big A, big A. This one had little a, little a. This one had big B, big B. This one had little b, little b. Remember, the spindle fibers attach, and these guys are going to get pulled apart. They're going to get pulled to opposite poles. And so let's zoom forward to what that's going to look like in metaphase 2. In metaphase 2, we're going to have 2 cells, and one is going to contain both of the paternal chromosomes. And the other one's going to contain both of the maternal chromosomes. Let's put our alleles on each of these. So this one still contains the big A. We're putting crossing over on the back burner for now. Big B, little a, little a, little b, little b. Spindle fibers attach. Remember, these are in separate cells. And then they're going to get separated. And what do we end up with here? We end up with, at the end of telophase, we end up with 2 haploid gametes. And with the end of this, we end up with another 2 haploid gametes. Now let's put our alleles on here and see what this means. Because remember this individual, what were the different gametes that could be formed? Remember, big A could be with big B or little b, right? So Mendel's law of independent assortment says big A will end up with big B just as often as little b. So we had this gamete, we had this gamete. Little a can end up with big B, little a can end up with little b. Right, so these 4 gametes are produced in an equal proportion. Okay, let's put our gametes on here. What do we have? Big A, big B, big A, big B. So we've got 2 AB gametes. And we've got a little a, little b, little a, little b. Well, wait a second. So we've got this gamete, and we've got this gamete, but how did we get these lines? So this one would be 2 gametes. Oops, whoa, something just happened here. I'm not sure how to get out of here. 1 second. So we generated our big A, big B gamete and our little a, little b gamete, but where do the other two come from? That has to do with the chromosomal basis for Mendel's law of independent assortment. And that has to do with the fact that the chromosomes, when they align on the indicator, do so randomly. So this alignment here is just as likely as, say, let's say the parental one is on this side during metaphase 1, and let's say the parental for the other was on that side. And then here's our maternal. These are synapsed together, even though they don't really look like it, but they are paired up. And look what happens here when these get separated. Spindle fibers shorten, they go to opposite poles, and entering into meiosis 2, one cell contains the large parental and one contains the small maternal, and vice versa. Now these are going to get pulled to opposite poles. And you can see where this is going, hopefully. And what do we end up with? We end up with 4 gametes. 2 of them contain the small, small blue chromosome. So there's 2 of your gametes like this, and the other 2 are like this. Assign the alleles to each of the chromosomes, and what do we get? So this red one had little a, and this had a little a, and this one had little b, and this one had big A, I'm sorry, big B, and this one had big A, big A. So this gamete is the little a, big B gamete, 2 of them like that, and then big A, little b, big A, little b. So it has to do with the arrangement. So Mendel's second law, the law of independent assortment, is really due to the chromosomal arrangement at metaphase 1, and that it's random. It's random. They're independent events. So there's nothing that links together the paternals and the maternals. The way they arrange on the metaphase plate is completely random. Therefore, in any one meiosis, you only get 2 outcomes. But if you take all the meiosis in the organism over its lifetime, what you end up with are an equal distribution of AB gametes, little AB gametes, little a big B, and big A little b, in a 1:1:1:1 ratio. Now, Mendel's first law, the law of segregation of alleles, that has to do with separation during anaphase, right? So the alleles gets separated when these chromosomes get pulled apart. Now, if there's a crossing over event, which we're going to talk about in the future, it's separation in anaphase both 1 and anaphase 2 if there was any crossing over. But we're going to separate our alleles, and different genes are not linked together because they line up differently, independently, randomly on the metaphase plate during metaphase 1.